So hello everyone. Um, I am Sourim Banerjee. I am a 2016 JB scholar and your host for this evening. This is our second Young Scholar Exposure event uh, for this year, and we have two special reviewers today, Dr. Sitabro Sinha and Dr. Atanu Kumar Datto. Our first reviewer is Dr. Sitabro Sinha. Dr. Sinha is a JB scholar of 1989 and currently a professor of theoretical physics and computational biology at the Institute of Mathematical Sciences in India. He received his bachelor's from St. Xavier's College, Kolkata, and master's in pure physics from the University College of Science, University of Calcutta. He received his PhD from the Indian Statistical Institute, where he was affiliated with the Machine Intelligence Unit. Later, he was a postdoc at the physics department of Indian Institute of Science and the Medical College of Cornell University, USA. His research interests are in complex systems, nonlinear dynamics, computational biophysics, system neuroscience, econophysics, etc. Thanks a lot, Dr. Sinha, for being here. Our next reviewer is Dr. Atanu Kumar Datto. He is a JB Scholar of the Year of 2001. He is currently an assistant professor in the Department of Biochemistry at AIIMS Kolani. He did his MBBS from Bardan Medical College and his MD from PGI-MER Chandigarh in Biochemistry. He did his postdoc from Christian Medical College and Hospital, CMCH, Vellore. He is actively working towards setting up a publicly funded diagnostic human genetics laboratory for Eastern and Northeastern India. Thank you, Dr. Datto, for joining us today. So before introducing our speakers, I like to describe the format of today's event. So each speaker will get a total time of 20 minutes. Then we'll take some questions from the audience. Audience can post the questions in the chat box and I will read it out. After that, we can proceed to the valuable opinion of our reviewers. I will introduce the second speaker of this evening, Monisha. So Monisha is a JBE scholar of 2019 and a final year undergraduate student at the Department of Life Sciences, Presidency University, Kolkata. She had completed her schooling from St. Teresa's secondary school in the year of 2019 and is highly indebted to this institution for not only making her fall in love with the nuances of biology, but also inculcating in her the values of life, which are so precious and rare. She was selected for the Science Park course in cell biology during 2021, which was an initiative by Harvard University and Lakshmi Mittal and Family uh, South Asian Institute. Um, as a biologist, she wishes to create a brave new world full of hope and possibilities. Monisha, please, whenever you are ready.
Yes, should I share my screen now?
Yes.
So today I will be speaking on NO or nitric oxide, which is a signaling molecule in biology. It was not known until the uh, until 1970s, but now it has been characterized as a signaling molecule. So first, for NO to act as a signaling substance, we need to synthesize NO. For synthesizing NO, we require a substrate and an enzyme. The substrate for NO is L-arginine and the enzyme is NOS or nitric oxide synthase. Nitric oxide synthase exists as a dimer and each monomer is structurally and functionally divided into two sections. First is the C-terminal reductase, which has NADPH, FAD, and FMN. And the second section is the N-terminal oxygenase. The N-terminal oxygenase is, it has site for interaction with the heme moiety and the cofactor BH4, or it is also known as a tetrahydrobiopterin moiety. And it has a interaction site for the substrate L-arginine. And in between these two sites, we have it has a molecular switch, which is the calmodulin. Now, electron flows from NADPH to FAD to FMN and finally to the heme moiety. The electron is used to reduce iron, to reduce iron to the ferrous state so that it can bind to oxygen, and after it binds to oxygen, the L-arginine can be transformed into L-citrulline and NO. So the calmodulin binding site is the most important site, because without this site, the transfer of electrons wouldn't have been possible. Now, we have three types of NOs in our body. First is the NNOs. The NNOs stands for the NOs which are secreted these NOs are secreted in the neurons, and their activity is associated with calcium. Without calcium, the NOs are not able to function. The NNOs functions as a transmitter in the brain. The second type of NOs is the INOs, or the inducible form of NOs. These second type of NOs are the most interesting NOs, because unlike NNOs and ENOs, INOs, it doesn't require calcium and it is not constitutively present. It is only synthesized in response to certain pathological conditions. So the third type of NOs is the ENOs, or the endothelial NOs. These type of NOs are also calcium dependent, and they are responsible for insulin secretion and for maintaining vascular tone and for regulation of cardiac function. I would like to throw some light on the second type of NOs, or the INOs. This is the typical structure of INOs. And as I had mentioned earlier, INOs are calcium independent, and their synthesis is regulated at the in the nucleus, or at the formation of the RNA. The production of INOs is in response to the immune system. When the immune system is activated due to some foreign bodies such as bacterial endotoxins or LPS, which is the lipopolysaccharide, and also via cytokines. So when the macrophage and the immune cells of the body, when they see some foreign material, in response to that, they generate the cytokines. And these cytokines act as signaling molecules and they help in the synthesis of the INOs. The NO, which is synthesized via the INOS, helps in formation of certain T cells, which helps in immunity. So we will now talk about how the interferon gamma, which is a cytokine, how it activates the INOs. When in response to some foreign invasion, interferon gamma is secreted. It acts as a signaling substance to the interferon gamma receptor, as we can see here. So the interferon gamma receptor has two types of receptors. First is the IFN gamma R1. Upon ligand binding, that is, when interferon gamma binds to the receptor R1, the R2, which is associated with R1, it transphosphorylates and it functions by oligomerization, and it recruits the JAK1 and the JAK2. The function of JAK1 and JAK2 is to phosphorylate the intracellular domains of R1, as we can see here. And after R1 is phosphorylated, STAT1 can bind. Upon binding of STAT1, they form a homodimer, and the homodimer is also known as gamma activated factors. So these activated factors then translocate to the nucleus, where they bind to the DNA. They bind to specific sites which are known as the gamma activated sites. And the gamma activated sites have promoters of interferon stimulated genes. In response to such binding and activation, we have the generation of a transcription factor known as interferon regulatory factor 1, or IRF1. IRF1 now binds to the DNA and it facilitates the transcription of interferon stimulated response elements. Now it facilitates the formation of the secondary response genes, which is INOS. And when the enzyme INOS is being formed, it can aid in the formation of NO. So this is one way by which INOs can be formed in response to infection. Next is the role of LPS, or polysaccharide in the formation of INOs. So lipopolysaccharide, it functions as a signaling substance and it binds to a receptor known as toll-like receptor, which is a component of the innate immunity. The toll-like receptors, the toll-like receptor 4 acts as a receptor for lipopolysaccharide. Upon binding of LPS, the receptor functions by recruiting an adapter protein known as MYD88 or myeloid differentiation factor 88. This works as a reaction center and it recruits many other downstream effector molecules such as IRAC. IRAC is a kinase which phosphorylates TRAF6. TRAF6 then recruits MAP kinase, and through a series of phosphorylation, the inhibitor of the NF kappa beta or the nuclear factor kappa beta, the molecule which is an inhibitor to NF kappa beta, MAP kinase helps in phosphorylating that, and after phosphorylation, the inhibitor is removed and the NF kappa beta is activated. Again, it translocates to the nucleus where it binds to the DNA and aids in the formation of INOs. And once again, when INOs is formed, it helps in the formation of NO. And NO helps in the generation of immune cells. It helps in the generation of T lymphocytes, which can act as which can defend the body from infections. So the NO is the smallest known signaling molecule, and it actually connects the innate and the adaptive immunity, and NO has been used in various medicinal purposes to cure certain disease, but elevated amounts of NO is known to cause autoimmune diseases. So even though NO is an enigmatic molecule because it can be used as a cure, and it is also a cause of disease. So I would like to end my talk here and I can answer questions.
Thank you, Monisha. It was a very nice talk. I will ask the audience if they have any questions, please unmute yourself and you can ask Monisha about that.
Could you help me how to stop sharing? Okay, I found it.
There is an option.
Yeah, I found it. Thank you.
Okay, so I guess I can go with the reviewers. I'll ask Dr. Sinha, please give your opinion about Monisha's.
Since I already went first in the earlier round, maybe Otonu can go first, and then I'll
Yes,
follow.
yes, definitely. Yes. Please, Doctor.
Thank you, Sinha sir. So, Manisha, it was a very good topic that you have selected. This nitric oxide is indeed a very important molecule, both biologically and for medicinal purposes also. Uh, 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 since you are just beginning your career as a biologist or a researcher, so definitely there is a long way to go for you. And but I some of the things I found may help you to improve your presentation in future when you present. So one thing is that the abstract that came with the email. So in that you have written it very well. The initial paragraph that initially nitric oxide was thought to be a toxic molecule. It is present in cigarette smoke, but later on we found that it is a signaling molecule. It has so many purposes. So that you didn't cover in your presentation. So I thought if you in the initial presentation, if you just gave an idea. Also in the abstract, you have written that there are some scientists, 2 or 3 scientists you have mentioned, who had discovered the biology behind the nitric oxide, how they found that it is a signaling molecule. If you start with that, really that will draw the attention of the speaker, the listeners. Also, because you are more likely presenting like a seminar where you have researched a topic and you have come up with some interesting facts and you just want to share those. So because it is, then you have to streamline that. What are the things you want to present? So if you are giving a seminar, the topic you have not yourself worked, but you have just gone through the literature and you want to summarize the finding that you have got from the literature. So certain heading you want to divide your presentation. So initially, maybe one slide can be there where you will give the specific learning objectives. We call it SLOs. So maybe 2 or 3 points will be there. So what the learners would be able to learn from your presentation, at least 2 or 3 things that by the time you end the presentation, they should have some understanding of these 2, 3 topics. We call it specific learning objectives. Then you take those objectives one by one. So you divide your lecture like one learning objectives, then second learning objective, then third learning objective like that. And also once you start your presentation, there is something called a set induction. So set induction is that something, some interesting fact you can bring about that will seek attention of the learners for your presentation. So the cigarette smoking may be a very important topic, that nitric oxide is a toxic substance. It can lead to critical injury to lung and other tissues. But at the same time, it is a very important signaling molecule also. That can be very good set induction for your presentation. And then how you design your slides. So some of the images that we have presented, actually, some had very good clarity. Like one X-ray crystallography cartoon you presented of the nitric oxide synthase had got a very good resolution, but some other didn't have that much good resolution. So you have to select certain images which have got good resolution, and then once you are describing a pathway, so you have given some signaling pathway of the nitric oxide, how the signaling is there. So if you are presenting the whole signaling pathway to the listeners, then it will be very difficult. They might become confused to where to look at. And when you are presenting, at first you didn't use the laser pointer. So later on you started using laser pointer. So laser pointing is a very good. So you can go to certain parts of the image that you are discussing. The other way can be you can use you can cut and paste little portion of the signaling cascade, so that you can make it like a cartoon, and one by one it will come, and then you can discuss in one way. The other thing is that in the right hand side you have written down the major points. So those points need not be sentences. So you have written certain sentences. So in PowerPoint presentation, most of the time we don't write sentences. We just write bullet points. So only few words in bullet point you can write, so that you can cover maybe 5 or 6 lines maximum in a slide. Your slides are not very much cluttered, so number of words was less only, but you can only write some points, not the whole sentences like that. And then finally, when you have covered all those specific learning objectives, then you summarize that what all you have covered, and then what is the take home message from that. So I think you can next time when you present in your college also or in any other way, you can just try to modify your presentation like that. So initially there is a set introduction, then there is a specific learning objectives, then a summary slide, then take home message like that. So you can try doing that. So these are my suggestions.
Thank you so much.
Thank you, Dr. Datta, for your views. Please, Dr. Sinha, please give your opinions.
Yeah. Okay, Manisha, can you hear me?
Yes.
Okay, good. Okay, so first of all, Manisha, I really appreciate the enormous amount of work that must have gone behind, collecting all this information. So it was extremely, full of, lots of details. So I congratulate you for that. Great work. Also, I quite like the way, you kept text to kind of a minimal in your slides, because, sometimes we have this tendency of, and I've been myself guilty of that, putting a lot of text in slides, and the audience, starts reading it, and then, you transition to the next slide, and then, like the audience is left halfway reading your thing, and so it's good that you really had minimal text. Okay, so that I really appreciate. Now, Atanu has already mentioned quite a few good points. So I will start with I kind of reiterating them, but in a different way. So see, I kind of like to view when I'm giving a talk as if I'm telling my audience a story. I think of myself as a film director. So when you're a film director and you have something to tell your audience, the primal rule is you don't just show them scene after scene after scene, right? There must be a natural way in which one sequence must flow into another. One scene must flow to another. The audience should not be left scratching their head. Okay, I mean, fine, I understood that part, I understood this part, but what's the connection, right? So, of course you can use jump cuts. Movie directors use it a lot, but jump cuts, again, should not leave the audience thinking, okay, what happened just now? Okay, you should give the audience enough clues as to why you did the jump cut. Meanwhile, okay, or at that time there was this going on, right? So you should give this, kind of a, so you structure in your mind. Think of yourself as Satyajit Ray, or if you prefer, maybe Hitchcock. Okay, you of course try to be Godard, right? But then the problem with Godard is that 90% of your audience would probably leave the audience, cinema hall without understanding what's going on, right? So once I kind of saw a wonderful kind of a clip on making movies, where the person who was taking this class on movie making said the primal rule of movie making, whether you're making a documentary, whether you're making a 5-minute short, or whether you're making a feature film, is no and then. What does no and then mean? It means that you should not structure your scenes such that oh, then X happened, and then Y happened, and then Z happened. Rather, you should go like this: X happened, as a consequence, Y happened, and therefore Z followed, and therefore this happened, right? So the consequences should be absolutely clear. So when you are kind of structuring your presentation, think of it like this: that can I actually show my audience this transition? And so the primary rule of talks is, especially when you are giving, what do you call, a presentation of as much facts as you had in your talk, is one, selection. What are the facts you would like to present? Because you can't present everything and expect the audience to get all of it. You have to select what are the most important facts. And the second is sequencing. In what sequence should you give those facts? All right. Now, I always found it very useful, and most talks in the Scholar Exposure events actually follow this, is to tell the audience right at the outset, what is it that you are going to tell them? Okay, so your title was beautiful. I had a lot of expectation from your title, so you said that unraveling the enigma of nitrous NO as an endogenous signaling molecule in biology. Now, but I was like, I kept waiting. Where is this, enigma going to come? But at the end, like when you showed your final slide, I thought that you were about to get into it, right? So you said that, that thus begins the story of the smallest signaling molecule. And I was thinking, okay, now the real show starts. And then you kind of ended the movie, right? So if I was an audience, I would like want my money back, right? That has cheated me, right? Was basically working up to the final, like real sequences, and then suddenly the end, right? Look at the sequel, right? So that I thought was a really, really going to be interesting talk. So I thought even like working up to it, because at some point you said something like it's the bridge between innate and adaptive immunity. So that I thought was the most catchy slide of your entire, and of course your final slide of your this thing, which probably should be put right at the beginning, right? That this is what the importance of NO is really all about. And also, as Atanu pointed out, that history is sometimes a very good hook to get the audience. So for example, you just mentioned that until 1970s we didn't know the importance of NO. Now it would have been nice if you had spent like maybe 1 or 2 minutes and having a slide with pictures of the people, the main players in this NO. So basically you're saying like, that we didn't know about this molecule until about 50 years back. And suddenly it is like one of the most darling molecules of life scientists. And my talk is all about why that is. So, the audience immediately gets to, like why you're giving this talk, because, throughout your talk, I kind of kept asking, why do I need to know this? Okay. So you had a lot of detail, which maybe, I don't know why I should invest my resources into thinking about. So you basically walked through all the signaling cascades, right? Now, I'm sure they are all important, but as an outsider, as someone who's not even a biologist, why should I even, focus on understanding that? So, and also a very important point we want to remember is that sometimes when you have a figure, it may not be worth your while to basically just verbally describe what is happening in the figure, because the audience already knows it. In fact, that's what the figure is there for, right? You don't really need to say that, look, X is here, or X goes to Y, goes to this. You could just, mention the key points that this is how the sequence works, and then the audience can, work it out. So when you're like, for example, if you're writing a comics, you see that, like people like Will Eisner and so on, they will advise you that your pictures and your text should complement each other. They should not just repeat each other. Okay. So when you're showing like the picture of, person walking down the street, and you write beneath that picture, so Harry walked down the street, that is kind of misusing the power that image plus text give you. Instead, if you said, walking down the street, Harry thought of murdering his wife, or Harry started thinking what Sally said to him yesterday. Everybody can see Harry's walking down the street, right? But we don't know what is going on in his mind. And text allows us to say that. So pictures and text ideally should complement each other, right? They should not just repeat, so one should not just repeat the other. Overall, I would say that you have to try to see how to frame this as a storyteller. Okay, so that would be, like my suggestion. I have absolutely no, what do you call, suggestion to you as for, the facts of your talk. There were huge number of facts, but I thought that if you really worked on your presentation, this would have been a fantastic presentation. All right. So maybe, like for whatever it's worth, you could keep this in mind for your future talks. Thanks.
Thank you so much.
Thank you, Dr. Sinha, for your views and opinion. And yeah, so like this, we can this event comes to an end. So if any one of you, the audience, can want to give any feedback or suggestion regarding this event, please go ahead. Yeah, thank you. Um, okay, so I guess there is no other comment. So we can end this event now, and thank you all the speakers, Santanda, Monisha, and the reviewers, Stabroda and Atanoda, for joining and give your time for this talk. Thank you so much.
Thank you.
Thanks for your feedbacks. Yeah.
Bye.
Bye.